Thank you everyone for joining today. This is the first ever Stromal Network Journal Club we're doing. We're hoping that as we do this more often, hopefully we'll be able to gain more traction. But regardless, yeah, just a couple community rules is one, be kind and respectful, and two is to share this with others because we'd love to have more people join our discussions. And I'm really excited today to talk to you about this paper, which is alveolar fibroblast lineage orchestrates lung inflammation and fibrosis. I chose this paper, one, because this work sounded really interesting. Lung fibrosis is a really big topic, and it comes from a really well-respected group led by Dean Shepherd, who has done a lot of work on integrin signaling. And so I thought this could be really great. And it's also really fun to see cell of origin studies with lineage tracing because you need to use a lot of mouse models. And when you have a big group like this, they have access to those models. So it's great. All right, so just getting right into it. As many of you probably know, pro-fibrotic fibroblasts, we often use alpha smooth muscle actin as like that marker for these fibroblasts. This group also said that we could possibly use CTHRC1, which is collagen triple helix repeat containing 1, as another marker. We'll get back to that later. But the bigger question is, what are the origin of these pro-fibrotic fibroblasts, specifically in lung fibrosis? A lot of different studies have looked by single-cell RNA-seq, but they haven't found anything very conclusive. A very important seminal paper published by Matt Buechler and Shannon Turley's group showed that with computational lineage inference, so using computers to try to figure out going backwards, that PI16 positive cells may come from, fibroblasts may come from adventitial cells. But the authors of the paper we're looking at today have published previous work using a COL1A1 GFP tagged mouse and kind of say they think it's more likely alveolar fibroblasts. So it's a kind of interesting topic of discussion between these two groups of where do these cells come from? Which is it? Is it the adventitial cells or these alveolar fibroblasts? All right, so the main mouse model that they use for the first few figures is these Scooby2 expressing fibroblasts. So what they do is they mark Scooby2 fibroblasts, which they call the alveolar fibroblast background. And then they knocked in Rosa 26 TD tomato. So what happens is when you inject tamoxifen into these mice, all of the Scooby2 expressing cells now express TD tomato. And yeah, it's really nice, easy model to use in that way. Oh yeah, there we go. And when you take these cells out, if you take the lungs out of these mice and then you look at them by single cell digestion and you use lineage markers to kind of get rid of non-fibroblasts. So you want to get CD31, CD45, EbCam, TER119, MCAM. All of these are different types of non-fibroblast cells. So it should be negative, lineage negative. But then you want your alveolar cells, which are SCA1 negative, CD9 negative, PGFR alpha positive. So this kind of this red area should also, they should be expressed by TD tomatoes. So your Scooby2 cells should be negative for all these ones, except for PGFR alpha, which is the classic, I guess, early fibroblast marker. Does this make sense so far? Cool. Stop me if I'm going too fast or if you have any questions at any point. Really happy to explain more. Yeah. So you have this great model. And what they wanted to see is kind of where it localizes in the lungs. So they took a lung out of the mouse and they did histology. So you can see this merge is the merge of autofluorescence, which kind of gives you the overlay of the lungs. You can kind of see this nice, like all these airways. TD tomatoes, of course, your alveolar fibroblasts. This is their previous collagen 1, COL1A1 GFP mouse model. I think they're just trying to show, and then it's kind of overlaid. You can, sorry, no, I'm wrong. This is the collagen 1, just GFP, just all total collagen. And they're trying to show here that your alveolar fibroblasts are kind of next to these areas of collagen. Yes. But also what they're trying to show in this bottom area is that these alveolar fibroblasts in normal lung homeostasis, they contribute to AT2 epithelial cell support. So these AT2 are alveolar type 2 cells. They're just the normal lung epithelial cells marked by pro-SPC in blue, I believe. Yeah. And they show that these red alveolar fibroblasts are kind of supporting these cells from the top and from the bottom, kind of like holding them. And so that in their normal, like injury state, they don't contribute to like the total collagen in the lung. Something else has made them because that's just the lung structure, but they do support these epithelial cells normally. And that's kind of their basic role. That's just also kind of known. So what they wanted to do now was to delete these Scooby-2 alveolar cells using Rosa 20, sorry, that's a Rosa 26, not 25, DTA. And so when you have a DTA on board, it deletes anything that's Cree-expressing when you inject tamoxifen. So in this model, when you have this Rosa 26 DTA, instead of the TD tomato, now you have no Scooby-2. And what you see immediately is when you induce with tamoxifen, a decrease in this pro-SPC. You can see here by representative IF, but you can also see here in this plot that significantly less AT2 epithelial cells. And here, further bringing that point out is, yeah, you also have less of these alveolar fibroblasts, of course, because you've depleted them. And they just use different markers to kind of say the same thing, which is EpCAM and positive MHC2 positive epithelial cells should also be these AT2 cells. You also see less of them. And then when you look through total qPCR in the whole lung, you see less Scooby2, which is what we expect. You also see less of this P4 alpha, which is the classic kind of non-differentiated fibroblasts, and the SFTPC, similar kind of marker. Cool. All right. And then now that they're like, yeah, this model works, let's induce some lung injury, see what happens. So bleomycin is a very common agent that's used to induce lung injury. They just kind of, I think they inhale it. And they want to see if loss of these Scooby-2 alveolar fibroblasts affects the response to injury. So this is kind of their model. You can see here that there's an increase in IgM in bronchial alveolar lavage fluid. I think, yeah, BALF. When they do saline, no difference. It shouldn't do anything. But when you do bleomycin, especially when you deplete the Scooby-2, you get more inflammation. And they also show that this increase in this kind of IgM is like the first cytokine that goes in when you have any kind of assault. So it's telling you that the immune system is getting activated. And around the same time, you also see neutrophil cell infiltration also increasing with bleomycin and insult. They also wanted to see different types of immune cell populations that they know exist in the lung and are responsive to lung injury. That includes gamma delta T cells, CD4 T cells, and innate lymphoid cells. But what's interesting is really all they see is a difference in increase in IL-17A, which is a specific inflammatory cytokine. Only in really these gamma delta T cells increased after bleomycin injury. Nothing really happens to these other two cell types. And yeah, I think this is an ELISA and Q. So yeah, P is qPCR, and Q is ELISA. Confusingly. But what was interesting is when you neutralize IL-17A, you can reverse these effects. So in blue, we have our classic Scooby-2 Cree-ER model, but just wild type Rosa 26, so no deletion. And then in the red, we have the deletion of the Scooby-2. So you see that decreases the body weight of the mice as would it be expected given the increase in inflammation. You also see increased more death, less survival. And then when you neutralize it using an anti-IL-17A antibody injected into the mice, you see a reverse of these effects. So they're trying to say in the end that one, Scooby-2 alveolar fibroblasts support these AT2 epithelial cells, but also loss of these alveolar fibroblasts in lung injury can cause IL-17A mediated inflammation. That's kind of the conclusion from this first figure. I know it's chunky. This was probably the biggest figure in the paper. So any questions about that so far? All right, okay. Moving on to figure 2. Now they wanted to actually look at what these fibroblasts do in response to injury. So to get a better picture of what these alveolar fibroblasts do once you induce injury using bleomycin, they collected single-cell RNA-seq from these mesenchymal cells of these lungs after bleomycin treatment at various time points. So day 0, which would be naive, and then bleomycin treatment, and then they collected the lungs 7, 14, 21 days afterwards. They took, and they also tried to specifically get mesenchymal cells using these lineage negative markers, so that most of what you would be sequencing would be these fibroblasts. In this UMAP, they initially got 11 clusters, but they say 4 of these clusters are specific to injury: fibrotic, inflammatory, stress activated, and proliferating. I have a better kind of breakdown of this here. So you can see in day 0, there's like none of these 4 guys, but then day 7, 14, 21, you see a lot more of these inflammatory in like this greenish, pink stress activated, blue fibrotic, and dark grays proliferating cells. And they also use specific markers for each of these. So fibrotic fibroblasts, they say, are higher in COL1A1, CTHRC1, and other fibrotic genes, whereas inflammatory like fibroblasts are CXCL12, SAA3, LCN2, and IFN-gamma are interferon mediated or have these genes. The stress activated is primarily an increase in P21, but they also saw increases in translation and stress related genes. And this proliferating cluster is mainly marked by high levels of MKI67, which is common proliferating marker. Yeah. And yeah, these don't appear in day 0, really. They're very, very little. So now they want to kind of see what the contribution of these alveolar fibroblasts are to each of these 4 subsets of fibroblasts in injury. So this plot is just kind of like a baseline. So like I said before, in this initial screen, they took the cells out, but they didn't take them out based on TG tomato. They just took them based on this lineage. So just to break it down, CD31 is endothelial or vascular, CD45 is immune, EpCAM, and they don't have the other one. EpCAM is an epithelial marker, and TER119 is a red blood cell. So they're trying to get rid of anything that isn't these 4 types of cells, most of which should be fibroblasts, but not all of them are TD tomato. So they did want to specifically kind of understand how much of these alveolar fibroblasts, especially the ones contributing to injury, were of the Scooby-2 background, which would be TD tomato positive. And they say around 75%, which is like quite a bit, because it's up to this study, there's not been too many studies that have shown from lineage tracing, which is the only way to really for sure show that like this is where a cell comes from, that this is where these cells are coming from. So this is really interesting. So yeah, they show that at steady state, around 75% of all of these different types of alveolar fibroblasts come from this TD tomato marked by Scooby-2 cells marked by TD tomato. And 70 to 80% of fibroblasts are of alveolar origin from every single one of these 4 fibroblast subsets, and this kind of holds over time. So no matter what time you took it from, almost all of these cells come from this cell of origin. All right. This is like a slight kind of like a detour. They wanted to, I guess reviewer number 2 was unhappy with the bleomycin model, and they're like, use a different injury model. So they tried intratracheal silica installation, which is another common way to induce lung fibrosis injury instead of bleomycin. And the question is, do alveolar fibroblasts contribute to lung fibrosis? And these other models, the answer is yes. Yeah, you see silica induces TD tomato. All these cells are there. Also high in PGFR alpha. You see these SAA3, LCN2 are both inflammatory fibroblast markers. You see them increase in the silica induced. Wow, that looks so similar to saline. Silica, not saline induced. Same with all these profibrotic markers. Okay, back from the segue. Now moving on to figure 3. Now the question is, we know that most of these fibroblasts in injury, lung injury, come from this Scooby-2 positive alveolar fibroblast background. How do they become, how do these alveolar fibroblasts from naive become inflammatory or fibrotic? So they do pseudotime analysis, which is pretty much when you take the cells from day 0, then 7, 14, 21, and you want to see what clusters from what. So it goes from alveolar, and then does it go to fibrotic or inflammatory, etc. They show that with the pseudotime analysis that 0 being kind of like the earlier time point, which will coincide with this alveolar region. As you move forward in time, pseudotime, you go towards this green region, inflammatory fibroblast, and then go down into this fibrotic region. Sorry, I should mention that they reclustered just based on these 3 types of fibroblasts, just to make the pseudotime analysis a bit more straightforward. And you can also see this in the expression of different genes that mark different types of fibroblasts. So you can see this In yellow, we have these alveolar fibroblast markers. They go down with time, pseudotime, but then the inflammatory markers go up, then go down, which indicates that it's becoming inflammatory, and then it goes down when things become fibrotic. Yeah, similar idea down here. So we know that we can kind of induce these inflammatory or fibrotic phenotypes using extracellular cytokines, IL-1 beta, TNF, or TGF-beta. So IL-1 beta and TNF are both inflammatory cytokines that are known to exist in the lungs during early inflammation and injury. And so they use both separately, but they really only found that IL-1 beta induced this expression of SAW3 and LCN2, which are the inflammatory markers. When you do TGF-beta, you kind of see the opposite. You see a slight increase in colon alpha 1, but you also see a drastic increase in CTHRC1. They have to have a better word for this one. So hard to say, CTHRC1. This bottom image is from extended data figure 7. I just included this one because I thought this was interesting. They took these primary alveolar fibroblasts. They did a first simulation with any of these 4 conditions. And then they did a second stimulation with TGF-beta or IL-1 beta. And what you can see here specifically, I guess, this third one is IL-1 beta, then TGF-beta. You can see that the TGF-beta kind of wins every single time. So whenever you have TGF-beta on board at the very end, you'll see higher levels of these fibrotic markers, colon A1, CTHRC1, but these inflammatory markers, SAW3, LCN2, you see that they go down, even though you know these are going to be induced in IL-1 beta conditions. So this kind of just shows that TGF-beta is like a stronger cytokine and has can override anything that IL-1 beta is telling the cells to do. So that I just thought that was interesting. It's a bit of a segue to later things they say in the paper. All right, moving forward with this control. Now they wanted to see if this kind of stuff that they see in this mouse pseudotime analysis would hold true in human patient samples that they had from human lungs. So they had 2 different sample types. They had idiopathic pulmonary fibrosis or IPF lungs, so classic fibrosis in like patient lungs, and they also had scleroderma lungs, which is, I believe, a type of lung fibrosis disease as well. And when they do a similar single-cell RNA-seq analysis, what they found was kind of interesting. They see this classic alveolar fibroblast type, but then they see 2 distinct inflammatory types, this light green, this dark green, and then the same fibrotic types as well. And they had control lungs that didn't really have any fibrotic cells, but they had some of these inflammatory cells, but mostly just alveolar fibroblasts. And when they do pseudotime analysis on these cells, they also see that you get this alveolar to inflammatory to fibrotic idea from the pseudotime, similarly as they saw it with the mice. Now they wanted to do it by histology just to prove to people that this is like actually what's going on. So you take the lungs, you look at them, and they see a similar thing, like these markers that are known to be associated with inflammatory or alveolar adventitial cells are all here. The ITGA8, they just included it to just exclude the adventitial fibroblasts because they're like, we don't want to look at it, but it's going to be there, so that's why that's there. But these other markers are here for all of these ones, and as you can see, I guess so what they do is they take these sections and then they kind of put it back onto the single cell object. So you can see all this colon A1, CTHRC1 positive areas are indeed in your fibrotic areas. SFRP2, CCL2 are both in your inflammatory regions, whereas this SFRP4, CXCL14 are more in this more, I think it was inflammatory 2. Yeah, inflammatory 1, inflammatory 2. Yeah. And finally, this is the actual histology inside of the lung sample to show you kind of where these different cells reside. So in IPF, what's known is that when you have fibrosis, there are these fibrotic foci, like strong regions of fibrosis, but around that region, you should expect inflammation. And that's exactly kind of what you see. So you see here an area of fibrosis denoted in yellow, that yellow area. You see high levels of CTHRC1 and colon A1. Kind of next to that area, you have inflammatory 1 fibroblasts, CCL2 ITG8 positive, SFRP2 positive, and this inflammatory 2 kind of on that border. which is kind of nicely shown in this diagram here in P. Yeah. All right. So in conclusion, from pseudotime analysis in both mice and humans, you see that alveolar fibroblasts kind of go through naive, inflammatory, fibrotic, this kind of pseudotime. And what we also found out is that TGF-beta wins against IL-1 beta in culture. All right. So now I alluded to this earlier before, but they wanted to look at specifically CTHRC1 positive fibrogenesis. They show that in their paper and in other papers, other groups have shown that this is a good marker for lung fibrosis of the cells that secrete the most amount of collagen. So using CTHRC1 pre-ER plus T tomato model. So now they have a new mouse model. You're inducing T tomato expression onto CTHRC1 when you have tamoxifen. So all cells that express CTHRC1 will start to express T tomato when you add tamoxifen. Just as a proof of concept, you see here that yeah, when you induce injury, they're all there. They all appear. Similarly, when you have this TD tomato model and again, you induce injury, you can see increase in COL1A1, CTHRC1, and osteopontin, which are all fibrotic markers. All right, so another thing to note about these fibroblasts and just about fibrosis in general in the lung, apparently in the lung, fibroblasts migrate from this basal lamina, which is denoted by collagen 4, so just like this general structure, into the alveolar airspace to create these fibrotic foci. So you can see in this untreated group, you have these really nice clean areas of like alveoli. Like when you look at a textbook, this is kind of like what you see. And then when you induce bleomycin, you can see that there's like a blurring of these edges and these fibroblasts are moving from that basal lamina and the collagen 4 into this collagen 1 region. From like these nice areas in pink to this like everyone going to the middle. This middle one is that Tsukui 2 model they had, so all alveolar fibroblasts. But you can see here really cleanly, in fact, that CTHRC1 specifically is well colocalized with collagen 1 regions in disease. And you also see no collagen 1 in this untreated model, which is kind of what you expect. And so what they're trying to show here is that these CTHRC1 cells are moving from these areas of these untreated areas into areas where they would start to secrete collagen. And they show this in F, where you show the distance from T tomato, I believe, to your area of fibrosis. Sorry, from collagen 4, which is where they came from. So they're showing that these cells are more migratory than just any of the alveolar fibroblasts. Yeah, sorry, G is the one where you show an increase in association of collagen 1 with these T tomato CTHRC1 cells over the just general alveolar fibroblasts Tsukui 2 cells. Okay, so now they want to do that fun thing where they get rid of the marker that they've just induced into the mice. So now they add a DTA again. They had some troubles with this model, and they admit that very plainly in the paper that they couldn't quite get rid of the TD tomato in these mice. So you kind of have a mixed population of TD tomato plus DTA. Yeah, so they just say it upright. But you do still see decrease in TD tomato with the DTA in this like gray. It should go all the way down if it worked fully. So that's how you know that the model didn't fully work. But you do, in fact, see CTHRC1 go down and like a decent amount. And then when they induce injury with bleomycin and you have this kind of DTA, like kind of you get rid of CTHRC1, you do see a decrease in hydroxyproline, which is a marker of processed collagen. So from here, we can sort of say that CTHRC1 does contribute to creation of fibrosis. And yeah, that's kind of the conclusion of this figure. CTHRC1 contribute to de novo fibrogenesis. They are highly migratory, move from this collagen 4 into this airspace, and they create collagen 1 and are accurate markers of this fibrotic fibroblast phenotype in lung fibrosis. All right. Any questions here? Any comments, concerns?
I just have one thing actually about the last figure.
For sure.
Just make sure I didn't miss anything. Like, I wonder if there's any temporal connection between inflammatory 1 and inflammatory 2.
That's a great question.
There.
And it was like the human one?
Yeah, that's right.
Yeah, that's a really good question. I think when you look kind of at the pseudotime, it's not so clear. And I mean, pseudotime also has its flaws in that, like, you can't quite say that this cell becomes that cell. You can just say that this cell looks quite like the cell based on the transcriptome. So it's a very good question and something that is definitely worth looking at. Yeah. And they do say that these differences in the fibroblast gene expression is highly correlated with induction of different types of cytokines. So it's probably, it could be that they go one into two, or it could actually be that they go separate depending on how these cytokines react in space. Yeah, so that's a great point, great question. Yeah. All right, moving on to the last figure, figure 5. So if you get rid of these fibrotic fibroblasts altogether, what happens? So they go back to these Scooby-2 cells because we know that these are our alveolar fibroblasts, and they wanted to deplete TGF beta R2. So this is the response to TGF beta that they would be able to have. And because these alveolar fibroblasts can become any of these cell types, that's why they chose these. And so when you do a conditional knockout of TGF beta R2, you should pretty much kind of skew towards those inflammatory-like cells. And also see what happens when you can't kind of continue in fibrosis, respond to TGF beta. You see a drastic decrease in collagen 1 in the lungs, just kind of upfront right here with bleomycin-induced injury. You see a decrease in collagen 1 in your bleomycin-treated lungs, as well as a decrease in hydroxyproline, which is, again, processed collagen. They also see this decrease in body weight that happens to these mice, so they're getting sicker. And you see an increase in inflammatory markers, SAL3, LCN2, CCL2, CCL7, and a decrease at the same time of fibrotic markers, colon A1, CTHRC1, osteopontin. And then you also see an increase in IgM in the bronchoalveolar lavage, so just like the bronchial fluid, which again, IgM is that primary cytokine that comes in when you have attack or afferent to like any system. So you can see that with this TGF beta R2 conditional knockout, when you induce injury, you get a higher inflammatory response and you get sicker mice. So as well, you actually see an increase in monocytes. So in this extended data figure below, you see SAW3 in pink, which is inflammatory fibroblasts, CD68, which is monocytes, and you see this association of these cells together. And then in H, this is figure 5H, you actually do see an increase in monocytes as well in these areas, as well as an increase in all these inflammatory markers, as well as the total area of monocytes. So lots of stuff going on, a lot of monocyte recruitment. They also noted that back here, CCL2 and CCL7 are monocyte recruiting cytokines. So that kind of makes complete sense. So yeah, when you take out TGF beta R2, so the cell's ability to respond to fibrosis, they kind of get stuck in this inflammatory state. But this kind of shows that if you kind of need TGF beta to induce fibrosis and kind of complete that cycle and to move out of that inflammatory fibroblast phenotype. And they have this really nice image here. It's like a graphical abstract of pretty much the whole paper. Yeah, so that's actually it. So in conclusion, alveolar fibroblasts Scooby-2 positive are normally there to support lung homeostasis as well as the epithelial AT2 cell function. However, they are the main contributor to fibroblasts and fibrosis following injury, and this is to all types of fibroblasts, namely inflammatory and fibrotic. TGF-BR2 conditional knockout, so when you get rid of these fibroblasts ability to respond to TGF beta, which is the main cytokine for fibrosis, you see that you can't get resolution from inflammation to fibrosis, which indicates that TGF beta is this very necessary thing that comes in that moves these cells from this inflammation state to a fibrotic state. And they also showed that CTHRC1 is a marker of late-stage fibrosis and is commonly found across like different human diseases. It's like in COVID lungs, it's in scleroderma, in IPF, and there's a few other they named there, but it's there in a lot of them. It's kind of an enigma because in this work that they show, it's not entirely what makes up all of the collagen 1 positive areas. And when you deplete it, you still do see some levels of collagen 1 and hydroxyproline, which is processed collagen. So this theory kind of does need a bit more work. And also they show, and they don't say this until the discussion, in the CT-HRC1 negative cells, you still actually see high levels of colon A1. So there are likely other cells that are contributing to these high amounts of ECM in the area. Yeah, that's pretty much the whole paper. Yeah. Any questions or comments? Or anything we want to go over again? Happy to go over any figures again.
Well, first of all, thank you for very succinctly presenting it. It was really nice. I guess one of the things that, let's say, one of the things that I really liked in this is that, you know, throughout, even with the stimulations, you know, the in vitro stuff, they really tried to drive the point home that, you know, TGF-beta is the, you know, the switch from inflammation to fibrosis. I think in terms of the wound healing response in the lung. And I think this point came across, albeit, for example, I think the CTHRC1 ablation was not the strongest point, as you mentioned, but they were very transparent about this. But even so, I think that this running theme throughout the paper was quite cleanly passed along, let's say. I liked it.
Yeah. Yeah, I also really appreciated that because I think personally, probably because I don't come from like a fibroblast background, but I always thought of cells, fibroblasts being like inflammatory like or fibrotic like, and this makes such a clean link between the two. And I think it's kind of been alluded to in other studies and other pseudotime analysis, but this is the first time I feel like where someone really shows with a lineage tracing model with like cytokines on top of cells that this is exactly what's going on. So yeah, I completely agree. It's a really nice point that they've made. It's really clean.
And I also like that you mentioned also, yeah, pseudotime has its flaws, let's say, as you said, but I really appreciate that on top of it there was like a clean lineage tracing, you know.
Yeah,
Sorry,
and unfortunately, we can't all afford to do lineage tracing models.
Of course. I mean, one could be so lucky, right?
Yeah, just to have one of these like mouse types, let alone, I don't even remember how many of them they had. They had so many. Yeah.
Yeah. So
Oh yeah, thank you for the presentation as well, by the way. I think you did a really good job describing the paper. I was actually just going to say exactly what you guys said. Like I found like the pseudotime, like the way they attacked the temporal nature of kind of fibroblast differentiation was really interesting because I'm like really new to kind of all of this, right? I like just started and yeah, I found this is a really good paper to kind of start me off looking at you know different ways to explore fibroblast differentiation and its role in something like lung injury. Yeah.
I think the other thing that I wanted to add on to all of this is that I really appreciated that they didn't try to cluster the cells further. Like a lot of single cell RNA-seq with fibroblasts, they get like 11 clusters in the like fibrotic or like so many of these inflammatory clusters, but they really zoomed all the way out and they're like, it's really only important that we just see inflammatory, fibrotic, like that's it. Maybe we see 2 inflammatory in the humans. That's it. Like it was really clean and they had really good sound biological reasons for difference in clustering. Whereas I think when you have other papers, and there's nothing wrong with it, I would say it's hard because a lot of times you get a bioinformatician to do the single cell and then a biologist to kind of do the analysis. So you don't always have biological reasoning for why you differentiate your clusters, but here it's very simple and very clean. And I think that's what makes the paper even more impactful. Yeah, for sure. Any criticisms of the paper or any like things you'd wish you saw more of? You can like take a look through. I think I had 1 or 2. I just need a moment to look through the images again. Yeah, and Caleb, you brought up a good point about the inflammatory 1 and 2. That'd be something really worth looking at.
Yeah, because I
Another
know
thing.
like, oh, sorry, no, go
No, go
ahead.
for it. Go for it.
I was just gonna say like, because obviously in the mouse model they just had the inflammatory, and I feel like, I mean, I could be wrong because but I feel like they didn't actually really do any kind of temporal study with the difference between inflammatory 1 and 2, and that may be because there isn't anything to be attacked there, so to speak. But I was just interested if they develop from each other or if they both kind of um differentiate at the same time, if that makes sense.
Yeah, for sure.
I mean, I'm sorry,
Go for it, Vasiliki.
I'm not sure if that has anything to do with it, but also, I mean, from the, you know, the UMAPs on scleroderma and IPF, I think the contribution of each inflammatory population in each of the conditions is different. I think scleroderma was contributing more to inflammatory set 2 and IPF to inflammatory set 1. So maybe even that could be contributing to this, to them clustering, differently, let's say. Maybe specific to the condition. I'm not sure, but,
Yeah, no, it's a great point. Yeah, I missed that, but that's a really good point that you see this very distinct difference in the IPF and the scleroderma. There's probably different like etiologies of how these diseases come about, and that probably plays a role in like how you get this difference in inflammatory 1 and 2. One other thing I want to say is that in the text, they mentioned that inflammatory 2 also had cells that had fibroblasts that had antigen presenting abilities. And in other groups, they talk about antigen presenting CAFs in like cancer. So maybe there's a similar role of these cells that have been kind of shoved into the inflammatory 2 area that might not so much have a role to play with like monocyte recruitment, but might have more of a role with like the adaptive immune system, like T cells and B cells and all that. So yeah, and it is something I would love to see more of, is this breakdown of this phenotype for sure. I think the one thing I missed it at the very, very beginning is I would have liked to see more and like justification for using Scooby-2 as your alveolar fibroblast lineage marker. They said it was from a previous paper. Then I looked at that previous paper and I couldn't find it in the main figures. And so I was like, that's a little weird. I don't know. It could just be like a very commonly known alveolar fibroblast marker. I'm just not someone who does lung disease, but that was like kind of my one thing. Just would want a little bit more convincing, just a little bit more.
I mean, you're right on the. I gave them the benefit of the doubt. I didn't have time to go back and look at the previous paper, but given the fact, I think you're right, especially given that their hypothesis that all of these subsets come from the alveolar lineage specifically, that is in contrast with the study with the PI16 positive fibroblasts. I think they could have done a better job at driving that point home, that this is a super specific alveolar fibroblast marker. So yeah.
Yeah, I in fact did look for it. I didn't look very hard, but I did look a little bit, and it was surprising that it wasn't like the first thing that pops out from any of these papers. But it's probably something they took out from their single cell, and it's probably one of their extended figures. But yeah, I thought this was just kind of interesting as well, that there's this kind of like difference, like everyone's just trying to figure out like where do these cells come from. It's like, feels like it's a very exciting time to be in this field. Yeah.
Yeah, even I just like literally just joining this kind of field, even I found like the heterogeneity between papers and how they differentiate is like it's crazy almost. Like there's almost no baseline right now. I mean, it's starting to form, but like it's so not black and white. It's so much gray area. It's really interesting.
It is, yeah, for sure. All right. Any other comments or questions?
I mean, we already discussed the, the DTA deletions. We discussed it on the CDHR-C1 CRE lines, but in the first one they did with the SCUBY2, there was a, what was it, 40%, up to 40% decrease in the positive cells, let's say, but which was fine. I just, not to be nitpicky here, but overall they said that in the morphology of the lung and stuff, it didn't cause much of an issue or trouble. And given that these are, the alveolar cells of the lung and that they were decreased, I don't know if that was because they didn't, sufficiently delete it, let's say. Or this phenotype only appeared during the bleomycin injury. But I'm nitpicking over here, if a phenotype is a phenotype, let's say, but since we're discussing, I think that's why I'm bringing it up.
No, a great thing to like bring up. And yeah, that's true. I didn't even think about that. They didn't quite deplete everything. So I wonder if maybe mice that had everything depleted got too sick to continue in the study. Like my one encounter.
I'm even thinking of whether, I don't remember how if they injected them, subcutaneously, whatever, with DTA, or,
I think it's like genetically. I don't really know how they do it. They like breed them. Yeah. So then the Rosa 26 DTA is a different mouse, and they cross it with a Scooby-Doo Cree ER. And then when you induce with tamoxifen.
For a moment, I thought it was the IDTR. Sorry, my bad.
Oh yeah, no worries.
Yeah.
Easy to confuse. One letter off.
Yeah. I was, but of course, in everything genetic, there's bound to be leakiness and stuff. But
as they come clean about in other ways too. Yeah.
But I like that I don't know, because as you said, with the silica model, some reviewer was, very intent on including everything. So maybe, so they don't exaggerate, let's say. I think they're taking it up to the point where it is believable and supported by what they're saying. So I liked that. But yes, just to be nitpicky about this ablation, let's say. That maybe if it was more, they would have seen something in homeostasis and not just during injury.
That's true. That is actually really interesting. A very good point you bring up. I didn't even think about that. Yeah. Yeah, because like you can see in like homeostasis, no change in like stuff in saline. I think you see more in the extended figures, but yeah. Yeah, only kind of a decrease in this AT2 cell marker, which is pro-SBC.
Also, I really liked your comment on that they're reasonable in what they cluster, because at first when I saw that figure with the 11 clusters, I was like, I misread it completely. I thought it was just the tomato positive cells, and I'm like, impossible to have 11 clusters of these. How? So I agree with you. They don't over cluster to over, increase the resolution to the point where there's no biological meaning no more. Yeah, so that was nice.
Yeah, as someone who's trying to hack away at single cell data, it's really refreshing to see.
Yeah, that's nice.